Hello and welcome. My name is Anaiz Ruiz, and today I will be talking about my summer research opportunity. This summer I had the pleasure of working with Chrissy Dominguez, who is a former Chafee student and is now at the UCR Department of Entomology working in the Herity Lab. This summer I dove headfirst into learning about my merids, or affectionately known as fairy flies. They are incredibly small wasps. I know when a lot of people think of wasps, they think of those yellow jackets or paper wasps, and they're pretty big, about an inch. Well, my particular specimen that I worked on this summer is only 0.5 millimeter long. Super small. My merids are typically egg parasitoids, and that makes them agriculturally and economically important as biological pest control. This family is made up of 116 known genera and over 1,400 described species. There is believed to be approximately 500,000 undescribed species, which is quite a bit. Now, this summer we worked on molecular phylogenetics. Molecular phylogenetics looks into DNA sequencing and where they genetically might match other specimens and where they might lie on the phylogenetic tree. To do the sequencing, we had to create voucher images. When we extract DNA from the specimens, they are put into a tube and it is done non-destructively, but there is a potential for them to get destroyed. So we take images in order to keep that in the database along with all the other genetic material and information we will have. Now, once we extracted the DNA, we amplified through PCR 3 different ribosomal gene regions. The data, once it was sequenced, was added to a molecular matrix, and statistical methods were used to place them on the tree. Once it was placed on the tree, it was pretty clear of where it might possibly belong to. And given its morphological features being so similar to other specimens within that group or tribe, it's possible that it belongs to the egg parasitoids that typically go after hemipterans. This experience was incredibly important in my entomology journey. When I thought of entomology previously, I thought, okay, you pin bugs, you identify them. Never had I thought they would be as small as this and as beautiful as they are under the microscope. I also never thought I'd be doing molecular work, of all things, to figure out where they belong on a phylogenetic tree. This was an incredibly important experience for me, and I hope that other people get to do this in the future, because these experiences really shape the way I look at the world now. And I see a small insect and I wonder, is it a parasitoid? What is it? What might it be? But because of all of this work that I've been doing, I will be continuing with the Herity Lab for the rest of the fall season, where we will be describing the new genus, and my mentor will be naming it, and it's going to be out there for the world to see. So I can't wait for that to happen. And thank you, Chafee, for this great opportunity, and the Herity Lab, and everyone else who was involved, Linda, Cynthia, and Sandra. Thank you guys for being so patient with me and being so helpful. And yeah, just thank you to everyone. It was a wonderful and unbelievable summer. Bye.